In today's lecture, we're going to be discussing the adolescent and aging brain, essentially continuing brain development, but looking at the later stages of it. And we'll discuss the challenges that the brain faces at each of these two stages, large stages of development. So as you learned in the previous lecture, the adolescent or the brain continues to develop well into the 20s and 30s. So the human adolescent brain is in a very immature state. There's a slow maturation of the human brain, particularly in those areas that handle executive function, things like decision-making and planning and behavioral inhibition. We talked about the fact that this was the result of that slow myelination of the prefrontal cortex. So the prefrontal cortex handles many of these executive functions, and the prefrontal cortex is one of those structures that is very last to develop. And myelination, which helps the brain become more efficient, continues all the way into the third decade of life. So the fact that the myelination takes so long produces a very immature brain during the adolescent period. So the consequence of this in terms of behavior is that you have very immature responses from the adolescent, such as impulsivity and risky decision making during this period, resulting in the adolescent likely putting him or herself in precarious situations that could lead to deadly consequences, an example of which is texting and driving. So Beatriz Luna and colleagues at the University of Pittsburgh explored some of the executive functions in children, adolescents, and adults because she wanted to compare what sorts of differences might exist between these three stages of development. So one of the tasks that they had subjects tested on required sustained focus on a goal. It required an ability to plan and carry out the appropriate response, and finally the ability to inhibit irrelevant responses that interfere with the completion of the task. This ability is known as response inhibition, and it is one of the many executive functions that the brain performs. So you might be wondering, like, in what situation can we use response inhibition? This seems like a very artificial scenario to put the subjects in. Well, how about when you're having a conversation with someone in a large room with friends and you end up hearing your name dropped in another conversation? What you need to do in that situation is inhibit your response to engage in eavesdropping on that other conversation and instead focus on the conversation you're having. So response inhibition is something that we have to engage in many, many times in our lives. So think about like needing to focus on this lecture and ignoring all of the other possible distractions that might exist. So responding to those other situations that might take your attention away from this lecture is your brain engaging in response inhibition. So researchers tested the subjects in an fMRI scanner. Remember, the fMRI, as we talked about in a previous lecture, is a scanning or imaging technique that allows you to look at areas of the brain that are active during specific tasks. So when they were in the scanner, they instructed them to stare at a light. Okay, very boring task. So when a stimulus suddenly appeared to one side of the light, either left or right, the subjects had to actually look in the opposite direction, right? Most of us would want to actually look at the light that showed up or look at the stimulus that showed up that was new, but instead what the subjects had to do was behave in the opposite way. So for example, if they were staring at this light and the stimulus appeared to the left, the subjects had to look to the right, okay? If they looked at the stimulus instead of looking away from it, it was considered an error. And because we naturally orient to new visual stimuli, as I just mentioned, success on this task required intense cognitive control to override that impulse to focus on the new stimulus that popped up. So results from this study can be seen here. So what you see is a stepwise increase in the mean correct inhibitory responses. So how successful they were at inhibiting their responses is shown on the left, and the subjects are divided by the age groups that the researchers were looking at. So you can see that children made many more mistakes than the adults, thus improvement on this response inhibition task as well as executive function improved through adolescence. So in determining what areas were involved in this function, remember they were in the fMRI scanner, those results showed that the anterior cingulate cortex and the dorsolateral prefrontal cortex, again both areas that are actually found in the prefrontal cortex, are needed to control the eye movements during this task. Interestingly, previous research on this topic showed that when teens were completing a task similar as adults, they actually had greater activation in the dorsolateral prefrontal cortex compared to adults. What this shows is that the teenagers could get to the level of the adults, but they needed greater brain effort than the adults. So this points to the fact that the teenage brain is not as efficient as the adult brain, and that could likely be the result of the fact that the prefrontal cortex is not myelinated. Remember, myelination speeds up the action potential. So if the neurons aren't myelinated, the efficiency of controlling these various functions is not going to be as good in the teenage brain as it is in the adult brain, and that's exactly what they found. So you want to think about the fact that the teenage brain exerted greater effort to produce the same result as what the adults did is analogous to trying to lift something heavy when you have little muscle mass. You may be able to lift that heavy object with lots of effort, but somebody with greater muscle mass is going to be able to more easily lift that object. And the brain myelination is the same way. The efficiency in the teenage brain is much less compared to the adult brain, and that's entirely the result of the lack of myelination. They have the neurons there. Those neurons are just not as efficient at communicating with each other as they are in the adult prefrontal cortex. So adolescents are also more likely than adults to engage in risky behaviors, and one reason for this is this tendency is a less developed ability to weigh short-term and long-term consequences. So researchers hypothesize that in the adult brain, the ventral striatum shown here, which is found in the subcortical areas, that's involved in processing of rewards, right? So the ventral striatum is actually part of the reward pathway and helps determine the value of rewards, appears to be hyperactive, while structures like the amygdala, which is involved in harm avoidance, and the prefrontal cortex, which is involved in regulatory control, both of them are less active in the adolescent brain. The result, unfortunately, is that you have an enhanced attention from the adolescents for more immediate rewards, like instant gratification, and less attention directed towards those long-term payoffs or consequences. So although risk-taking is often associated with adolescents than adults, researchers contradicts this idea that teenagers are these irrational individuals that are completely clueless about the potential harm that could result from the various behaviors they engage in. On the contrary, the logical reasoning ability of adolescents is actually similar to that of adults. There's a lack of evidence that it's the result of an underdeveloped prefrontal cortex. What it seems to be is that there's a mismatch between the maturation of different brain areas. So the normal trajectory of brain development involves maturation of the reward circuits, maturation of the limbic areas that occur much earlier than the maturation of the prefrontal cortex. So this mismatch, right, so the adolescent period, this is age, so the limbic areas develop much earlier than the prefrontal cortex areas, so then you have a period of time in life where the human child is going to engage in much more risky behaviors. So the fact that the limbic areas are much more developed, it's accompanied by this rise in dopamine. If you remember, dopamine is the neurotransmitter that is part of the reward system that allows us to derive pleasure from various situations. So given that the prefrontal cortex is not quite matured at this age, combined with the maturation of the limbic areas, it may help us better understand why the adolescents seek risky behaviors. It's entirely for the thrill, not because they lack an understanding of the consequences of their action, but rather they need that extra thrill for the same reason that they might be more likely to engage in things like riding roller coasters. So adolescents are not unable to understand the consequences of their actions. Rather, they are so seeking of exciting behaviors or exciting situations that they're likely to engage in those risky situations. So the adolescent period is also a time of onset for many mental disorders, and they include things like depression, schizophrenia, bipolar disorder, eating disorders, anxiety disorders, as well as substance abuse. Scientists have suggested that it may have something to do with brain maturation. So remember, most of you might think that, oh, brain development's done when you hit, 13 to 18. But in fact, the brain is changing in very, huge ways. So the adolescence is a time of dynamic changes in the brain. Any environmental challenges that occur during this period of time may negatively impact the developing brain, just like alcohol exposure or nicotine exposure during fetal development can negatively impact brain development during younger years. Any environmental challenges that could exist during this period, the adolescent period, can also impact the developing brain. This might be a reason why the stage of development is so much more susceptible to mental health disorders than any other stage of life. So we're going to change gears a little bit and talk about the aging or adult brain. So unfortunately for people like me, soon after the brain develops, there is a very short window of time before the brain starts to deteriorate. However, in the earlier part of the 20th century, Ramon y Cajal, you remember the father of neuroscience, asserted that once the brain completed its development, it was very fixed and immutable in adulthood, that it was a rigid, static structure. Fortunately, we have mountains of evidence that this is not the case. The adult brain, is quite plastic. It is not as plastic as it is during development, but it is capable of quite a bit of change, more than Ramon y Cajal would have thought. It is this idea that the brain is capable of change is the premise behind neuroplasticity. So the first scientist who demonstrated evidence of neuroplasticity was a biopsychologist pioneer, Carl Lashley. He observed the plasticity in the adult rhesus monkeys, and he did this in the 1920s. But given the belief at the time that the adult human brain was very rigid and static, many of his ideas were rejected by the scientific community. However, when William Greenough in the 80s of the University of Illinois investigated the effects of enriched environments in old rats, not just young rats, he found that they were just as effective, these enriched environments were just as effective at changing the brain in these older rats as the environments were to change the brains of younger rats. But people wondered if that was because, you're dealing with rats and they're not primates. So, here's some evidence that neuroplasticity exists in the primate brain. And this might, bother some of you, but they did this research in the owler monkey's hands. So under anesthesia, researchers amputated the third digit of the adult owler monkey's hand. So what they had done was they had kind of mapped out the hand into the brain, like what parts of the somatosensory cortex control various digits in the owler monkey's brain. And this is kind of how they mapped out the thumb is here, next to it is the second and third digit, and fourth and fifth. So they take up roughly this amount of space in the somatosensory cortex. So once they amputated the third digit of the adult, they waited about eight months to see if the brain had reorganized itself, right? So remember, they amputated the third digit, which is right here, but now the third digit no longer exists. Is the brain going to maintain this space for the third digit that doesn't exist, or is it going to repurpose and reuse the space for something different? When they mapped out the brain 8 months later, they found that the adjoining fingers, digits 4 and 2, took up more space than they did before when the finger was still present. So the brain repurposed the space that it once had for the third digit. It didn't just keep it there and wasted it. Instead, it repurposed it for the second and fourth digit. What was more incredible was that now digits 2 and 3 became more sensitive. They were able to detect more information, more touch information than they did before, and that's largely because they were now taking up more brain space than they did prior to the amputation of the third digit. So the next question was to determine how day-to-day experiences altered the brain's mapping. So, you're not going to find yourself with an amputated digit, but you can change how much you use your hands during the day. How does that impact the space that the brain sets aside for your digits? So again, still in the owl or monkey. So this was before training, right? They mapped out the digits before the training, and what they did was they stimulated certain fingers more than others, and specifically they stimulated digits 2 through 4 more than the thumb and digit 5. And what they found was greater stimulation of these digits resulted in them taking up a larger space in the brain. So digits 2, 3, and 4 had larger cortical areas compared to the thumb and digit 5. What's amazing is that we also know this is true in human beings. So musicians that use their left hand for greater manipulation, like violinists shown here on the right, had larger cortices, motor cortices, in the right hemisphere than their left. So remember, the opposite half of the brain controls the body. So if you're more likely to use your left hand as a violinist, it means that you're using your right hemisphere more. And that's exactly what they found. The size of the motor cortex of the right hemisphere was much larger in violinists compared to their left hemisphere, which controls their right hand. These violinists also showed a greater sensitivity in those fingers of their left hand than their right, regardless of handedness. So even if they were right-handed individuals, because they use their left hand so much to manipulate the violin and play the instrument, they showed greater touch sensitivity of their left hand than they did of their right hand. You might not have any experience with this, but I certainly do, and many of your parents and grandparents might experience this, but during normal aging, many sensory abilities change. So your vision gets worse, your hearing gets worse, and it might be that the sensory neurons are degrading, but it might also be that the neurons in the brain are weakening. In addition to sensory abilities, aging adults also lose their cognitive ability. Most adults experience a reduction in processing speed as they age. They're not as fast at calculating or thinking as the younger brain is. Post-mortem studies have shown, so this is after death, have shown that brain weight, size of neurons, synaptic density and dendritic spine, so the ability to communicate with other neurons, all decrease with age. So this doesn't bode well for you guys as you get older, but we'll talk about the ways in which you can counteract the normal aging process later on in the lecture. So there might be many potential causes of aging, but one of them that we can consider is a genetic decline. So telomeres are part of your DNA. They're repetitive structures, and they usually are found as caps on the ends of your chromosomes. What they do is they keep our chromosomes intact during replication. So as we age, we become these telomeres become weaker and therefore protect our DNA less, resulting in greater cell death. So if the DNA becomes destabilized in a neuron or any cell for that matter, the cell is going to not stick around and instead commit cell suicide and say like, oh, I'd rather not cause any problems here, let's just get rid of the cell. But the more it happens in the brain, for example, if more telomeres are degrading across many neurons in the brain, it could possibly lead to that cognitive decline that's typically seen in normal aging. So you normally see cognitive decline as the brain ages. But what's unusual is the cognitive decline seen in Alzheimer's disease. So in these individuals, unlike normal aging, the brain deteriorates at rates that are unusually fast. So initially, it starts with loss in short-term memory, and eventually it could progress to the point where the patients can no longer care for themselves. The kind of deterioration seen in Alzheimer's disease can be seen in this image over here. So you see huge shrinkage of the hippocampus. The ventricles are enlarged, right? So as you're losing brain space, you're seeing more space in the brain area. I'm sorry, as you lose more brain mass, you're going to see more space taken up under the skull. You also see shrinkage of the cortex, particularly cortical areas like the prefrontal cortex that handle executive function. So these brain areas, for example, like the hippocampus, they're critical for learning and memory and advanced cognitive function. These areas have the greatest neuronal loss. So what are the causes for Alzheimer's disease? So what we don't know at this time still, after 100 years since this disease was discovered, we don't know what triggers the disease. But we do know what ends up ultimately causing the death of neurons. Too much of these two characteristics eventually causes the death of neurons. The first of them is the amyloid plaque. So there are two things that are characteristic and are used to pathologically diagnose Alzheimer's disease: plaques and tangles. The plaques are the collections of beta amyloid plaques. So these are proteins. Beta amyloid is a protein, and for some reason that we still don't know, they end up aggregating and collecting and kind of clogging the outside of the neuron so that other neurons are unable to speak to it. So this is a synapse here, and because of the amyloid plaques surrounding the neuron, this neuron here is unable to communicate. As a result, lack of communication, this neuron can't receive the proper information that it needs to perform the function. The other characteristic are tangles. So tangles are the deconstruction of the proteins that are involved in stabilizing these train tracks or microtubules in the axon. So proteins, tau proteins, are normally attached to microtubules to keep them stable, and the microtubules are found in the axon. Unfortunately, for again reasons we have no clue about, the tau gets what's called phosphorylated. It gets extra tags placed on it. So these extra tags cause the tau to pop off the microtubules and create these tangles, these neurofibrillary tangles seen here. So two things happen. The microtubules become destabilized, and these proteins are critical for transporting materials through the axon. And then you have these neurofibrillary tangles, these bunched up proteins that are formed within a neuron, and they end up clogging the neuron from within, right? So the neuron can't function the way it normally needs to, and typically you find these tangles clogging the entire cell body of the neuron. So think about what the cell body does, and imagine that you've got like this meshwork of clogs of proteins preventing the neuron from doing what it needs to do to survive. So it shouldn't be a surprise then that this ultimately results in neuronal death. And if you have massive amounts of these plaques and tangles building up across the brain, you can see why the brain ends up looking like it does in Alzheimer's disease. You have massive deterioration of the cortical and hippocampal areas, and you have larger ventricles, and ultimately that results in decreased function in the patient. So negative plastic changes are what a researcher, one of the neuroscientists whose name escapes me at the moment, calls what happens during normal aging. He said that the signal-to-noise ratio, which is essentially our ability to pull meaningful information from all of the sensory information that we're getting in our world, gets worse. So as we get older, our brain finds it harder to pull meaningful information and not kind of get distracted in the sea of information. But to counteract these negative plastic changes, older adults can engage in plasticity-based training programs. So you've probably seen many of these programs being advertised. You know, Lumosity is one of them that does a really good job advertising for its program. But those types of plasticity-based trainings work to enhance sensory functions, processing speed, and memory. And what some researchers have shown is that these plasticity-based training actually appear to improve the aging population's performance in these areas. So it's not that if you train your brain with these, you can get better at everything. What you're doing is you're training your brain on those specific tasks, so you get better at performing those types of tasks. Exercise also seems to improve brain plasticity and function in the aging adults. And I'm going to share with you a study that was done that's not in your textbook, but I find incredibly interesting to illustrate that even into the 80s, the adult human brain is capable of change with something that's very easy to do and costs very little money, exercise. So in this particular study, they had 120 subjects between the ages of 50 and 80 years old. So these might be, your grandparents' age. They served as subjects for this experiment, and they had these subjects perform what are called baseline experiments. So they wanted to, baseline measurements. So they wanted to kind of measure what they were at the start of the study. So they had them go into an MRI and measure their brain size of an area. They had them perform memory tests. They had them perform fitness tests, and then took some blood to measure some protein levels in their blood. Then they grouped these adults into one of two groups, the aerobic exercise group and the stretching group. 6 months later, so again, they had these subjects do these tasks in the laboratory. So the group that was in the aerobic exercise group performed the aerobic exercise in the laboratory. Those that were part of the stretching group performed these exercises in the laboratory. So then 6 months later, they had them come back, measure their MRI, memory test, and fitness test. I should tell you that these participants engaged in these tasks, these two types of exercises, every single day for 30 minutes a day for an entire year. At the end of the year, they had them remeasure those characteristics that they did at baseline. So the MRI, the memory test, the fitness test, as well as blood values. And this is what they found. So they measured an area that you guys are familiar with called the hippocampus, and the hippocampus can be seen here in yellow. So they measured the size of the hippocampus and compared the exercise group to the stretching group and showed how the hippocampus changed over the course of the year. So you have two hippocampi, one on either side, left and right, and you'll see that the exercise group, the aerobic exercise group, had an increase in their hippocampal volume compared to the stretching group over the period of the year. And the only thing that really differed between these groups really was which type of exercise they engaged in. What's more incredible is that this wasn't just, grow the hippocampus to make it bigger. They had functional differences. So in this particular slide or graph that I'm going to show you, they compared or correlated the percent change in memory performance, remember they had them perform those memory tasks, with the percent change in the right hippocampus volume. So some subjects experienced shrinking of the hippocampus, some subjects experienced growth, and when they correlated this with the change that these subjects had in their memory performance, this is what they found. They found a positive correlation between the hippocampus volume and their memory function. So this suggests that the larger hippocampi that grew as a result of exercising resulted in these participants having better memory performance in adulthood. So this is one way in which we could potentially counteract the actions or the decline experienced with aging. So let's summarize this lecture. The human brain matures slowly on into the second decade, and as a result, the adolescent years are characterized by poor executive functions and more risky behaviors. Adolescent years are also characterized by the onset of some mental disorders that we discussed, such as depression and anxiety disorders. Although it's not as plastic as development, neuroplasticity continues into the adult brain. So as the brain ages, though, sensory and cognitive abilities decline, but a greater incidence of diseases like Alzheimer's disease is also evident at this time of life. Damaged telomeres may be one reason for the loss of function in aging, but normal cognitive decline, and I mean normal, right? So you can't counteract Alzheimer's disease, for example, with these. Normal cognitive decline can be counteracted with brain training programs or exercise. So that's it for this lecture. Don't forget to submit your reflection.